Greetings Buy 100 team and welcome back. We're hoping that this video finds you happy and healthy, even though we know that the semester is chugging along and you're doing a lot of work both in and out of the classroom. We're really grateful for your efforts and your attention, and reminding you here just for a quick moment that if you need help, we're very happy to do that. Come stop by and talk to any of us, visit office hours, drop us an email. We really want to be here to support you. And again, we're proud to bring you this content. Today's video here is going to make good use and build upon some of the ones you've already interacted with before, particularly the reading week, how to read a scientific manuscript. And that's always a daunting process. No matter what level of scientist you might be, I still find it very challenging to pick up a paper and really break it down thoroughly. We absolutely understand that and are hoping you're starting to feel just a little more comfortable doing this process. Today, and in this particular video, we're going to kind of give you an overview of how to process those manuscripts at a very critical level, at a very analytical level. And we're going to break it down into two pieces. The first is scientific peer review. We want you to understand the process for how scientists evaluate a manuscript before it's published. What is all that business end of the story? And the second is, as a reader, how would you break it down and learn whether to trust a result or question it? Learn how to think about the conclusions and what they might mean, and process your overall acceptance of a particular work and your understanding of it. So writing those down into particular learning outcomes, here's what we hope. First, with regards to the peer review process, we want you to understand the submission and mechanics of how a manuscript goes from the lab bench to the journal. And secondly, we want you to be able to use all you've learned about reading a paper with regards to its very particular anatomy in order to then identify areas of potential concern. And I think this is a very important topic in a world of headlines. If you just read a journal title, if you just read a headline without vetting the content, you might be misled a little bit. And we don't want that to happen. We want you to be thorough users and vetters of the scientific method before you will accept a result or a statement as true. Well, it's manuscript submission time. And if you're the scientists who just performed a lot of work to get here, well, it's time to celebrate. This is a big step, and it's cause for celebration. It really is, because did you know that it could take 7 or more years sometimes to go from initial conceptualization and design of an experiment to then troubleshooting many results that go with it, to building a story that eventually gets published, working with potential collaborators across continents, between different universities. Sometimes interdisciplinary work is involved, so you have to mesh chemists and biologists and physicists alike. That does take a lot of time, and it could take years to do. And that fact blew my mind when I learned it. So if you hadn't had any appreciation for how long it might have taken a particular group to go from designing aims and questions and hypotheses, all the components of the methods that you've learned that science is built upon, to then actually getting it into a journal, well, I'm right there with you. I hadn't thought about it much myself when I was in your shoes. And it was a very eye-opening one when you think about it. So we're going to give you a general overview of this process and sort of the business that goes behind it. And as usual, this is a snapshot of it, and we're not going to be able to cover every single consideration, but we hope this gives you enough to go from as you leap into other classes afterwards and into your careers. We want to have a nice appreciation for you of where to at least kind of start thinking about all this. So here we go. First things first. You wrapped up that last experiment, you put down your pipette, and it was time to start to write this story. And if you're doing it right, you might have been kind of writing it along as you were going from experiment to experiment. But you'll work with your fellow authors to choose which particular journal you want to submit to and follow the formatting guidelines of that particular journal. That sounds straightforward, except it can't necessarily be that all the time, right? And it turns out that every journal has slightly different rules, slightly different guidelines, and each journal has a particular set of rubrics that they follow for whether or not they want to consider publishing a particular paper in their journal. And so we're going to kind of go through some of those considerations with you, because not all journals are the same, and therefore not all articles from journal to journal are the same in their scope and in their findings. Impact factor, then, is actually a number that's assigned to each journal. There are various scales. The higher the number, the higher the impact factor. The more rigorous a scientific body is in order to have it published in that particular journal, that requires oftentimes really advanced methodologies and a very advanced mechanistic understanding of a particular topic, or moving a field in a big way in terms of its overall understanding of the biology or chemistry or whatever the topic might be. Maybe we learned something very big about human beings in the process. Those are all very impactful findings. Potentially it was a medical journal like the New England Journal of Medicine, and they did a big patient study with lots of controls and healthy and diseased people, and we learned something about, let's say, diabetes, and that could be a very impactful finding. In comparison, at the other end, potentially it was something that wasn't so broadly human-based, but maybe we understood a solid mechanism. That's great science. Please don't think that impact factor is an indication of the worth of the science. We don't think that's true. But at the lower end, that could mean simpler methods and a more just solid understanding of a mechanism from start to finish, and building upon the scientific knowledge in that way is, again, just as important. So we do not want you to think that impact factor necessarily means importance. But scientists do have to choose where they want to submit their journal, and so impact factor is an important part of that process and where to shoot for in terms of the scope of their work. Whenever you look at a paper, you see the author lines and all the people involved, and so the group has to make a decision about the ordering of people and how that works. And so journals have a very defined set of criterion for what constitutes authorship, from whether they were involved with the design and creation of the experiments themselves, to actually performing them at the lab bench, and then subsequently analyzing results and writing and reviewing the work. So you follow those journals' guidelines to determine who gets to be on that all-important authorship line up at the top of the first page, and then might also give you guidance on who would come at the end of the paper. Sometimes you flip to the back and you'll see an acknowledgments. Maybe someone helped with a particular design of something, but it wasn't significant enough to be in the primary authorship line, but you want to acknowledge someone for their contribution in some way. You have people on both ends there. I think it's important to remember that it's people doing the work, and when you look at the authors, you'll see one that's the corresponding author, and that person can be emailed from anything from, could you please even get me a PDF copy of your manuscript, to wow, you guys made a really neat mutant strain of bacteria, and I'd really like to have that in my lab. If I paid for the shipping costs, would you be amenable to sending it to me? Corresponding author can be contacted for any of those things. And you also see their affiliations, which gives you a really good idea of where the work was done. And that could be an important consideration for you, young scientists, if you're thinking about going into graduate school or trying to get work in a particular area of the state or different country. Knowing who does what and where is a key thing. And as you get involved in science, you'll see that it's actually a pretty small world. Every journal has very strict word and or page limits, and so that is a very important consideration, and it will become even more so later on in this video, as I tell you sometimes a paper is submitted and then returned to the authors, and the decision is please submit it somewhere else, and then you pick a different journal that has a completely different set of word and page limits, and so you have to reformat and rethink your story, and strict adherence to these word and page limits is a key for the publication of your paper. And of course, figures and supplemental materials. Journals have very established criterion for what data should look like, and we had a great data visualization module already. But sometimes you'll see a particular figure have multiple subpanels. And sometimes you'll see papers have a set of supplemental materials as well that add on to the paper's findings, but that aren't included in the primary set of pages. And so it's usually a separate file linked at the end. So after choosing which would go where, which figures and tables and other types of data would go into the primary body of the work, and which would potentially go as supportive documents in a supplemental materials, which actually also includes sometimes a more detailed description of a method. Now it's time to put that all together and get it submitted, and usually journals have an online submission process for that. Once you press submit, you breathe a sigh of relief. You have some fun with your lab. Maybe you ring a victory bell in the lab. Some actually have that. And then you worry and you start to get very nervous with each passing day because it's now time to wait. And no one likes waiting. Not for anything. Not for a bus. Not for a parking spot at Sac State. Not for anything. I don't like to wait. I'm very impatient. But now we have to do that, and the fate of your manuscript rests on the following couple of procedures. Your journal, once it goes through the online submission queue, is assigned to an editor that is a scientist in your field that knows your work and the scope of the methods and findings you might have there. And their job is to vet that and make an important decision about whether they think that whether or not your submission meets the standards and criterion is aligned with the impact factor of their particular journal. And this was surprising to me too. I had no idea about this business. I also didn't know that the editors and the peer reviewers that we're going to talk about in a moment are all doing this on a volunteer basis. And so this is not actually a paid position at a journal, which I thought it might be. This is typically done by scientists who are at universities or companies, and they're just very, very vested in having a high-quality science published to contribute to the body of knowledge in their particular field. So we wait for what's called editorial review. This process takes time, and hopefully not too much time. But here's what's under consideration. Whether or not the work meets the scope and purpose of the journal, and an evaluation of the overall scientific merit of the particular study. The novelty of the methods, how new they are, how important these findings might be, and the editor has a very, very important decision to make. They can let you know that they think, yes, this does indeed accomplish those two things, and so therefore I will send along your manuscript for peer review, which will take more time, or I will outright reject that manuscript and say, I'm so sorry, this does not accomplish those two things. And hopefully a good editor will provide you some feedback and some guidance if they do reject it, to help improve your study. I really believe that the purpose of all of this submission and review is to make the science better. And I hope that that's the approach by most, although it's not always. Some people just like to be critics. I always kind of think about that super negative Yelp reviewer who is talking about going to dinner with their BF, and they go on and on about all that, and I don't really care about that. I just want to see a picture of the food and just kind of move on from there. I'll decide myself. You don't need to be too negative there, but sometimes scientists are very, very negative during this process, and that could be super deflating. So hopefully a good editor and then subsequent peer reviewer, even though their job is to find and vet the scientific body of work, if they do suggest improvements, it's for the sake of improving it and not just to cut it down. So the clock is ticking. We hope that this editor reviews and makes a decision in a very speedy and timely manner, but sometimes that could take a little while. Sometimes it could take a month or two, hopefully less than either of those options, because the fate of the manuscript then takes more time. So if the editor decides to reject the study, says, "I'm so sorry, the journal thought your work was great, but not quite significant for what we try to do here," now we have to return to step one. We have to go all the way back and choose a new journal, and now maybe actually have to rewrite or reformat those findings, or maybe even do some more experiments. Perhaps the editor said, "Hey, if you do these things, you could resubmit to our journal." So you might have to just add a few more experiments and then resubmit to that journal. Or you might say, I don't want to do that. Maybe we'll just go to a different impact factor journal. If the editor said, "Congratulations, I think this does meet the criterion," then we'll go on to step 3 of the process, which is our next slide here. Oh, good. I'm so glad to see that I have to wait again. Yeah, so the next step is waiting once more for what's called peer review. And so the editor is going to assign your journal, your manuscript article, to folks in your field. And again, this is a volunteer service in most places, to either 2 or 3 scientists in the field. Depends on the journal. Sometimes there's 2 reviewers and sometimes there's 3. And believe it or not, this particular part is anonymous. So you, when you submitted the article, knew the name of the editor, but you don't know the names of the researchers performing the peer review. They do know your name and all the authors, though. So there is a bit of a conflict of interest there. And what I like is this new movement in science to have everything be not anonymous, but open. And so there's lots of journals now requiring reviewers to put their name on it and even have that name published on the article as a reviewer. And I think that's a great idea. I think more transparency and more understanding of who's who will allow to a more kind and thorough, actually, scientific review process, just as when you have to put your name on your reviews online for something. So after the peer review takes place, which again could take a couple of months or more, you have the following 3 decisions that might come to your inbox as you are constantly checking your inbox every single day, wondering about the fate of your work. The first is the most unfortunate type, and that's outright rejection. Potentially the peer reviewers thought, while it did meet the scope of the journal, there were flaws in either the methods or the study design or both. Potentially they thought you needed to do a significant amount of revisions and further experiments that they thought were just beyond the scope of this particular submission round, and so they rejected outright. If that's the case, you're going to have to return to step 1 and again choose the journal. You might hopefully get some very helpful feedback from those reviewers and decide, let's do those experiments and submit again to this journal. Maybe that takes you another year or even 2 years to do, or longer, or shorter, but it could take a long time. Or you can decide to submit to a different journal, and that might again require some rewriting and revision. This is the second time that could happen. Another potential outcome of peer review is one for medium level of celebration, acceptance. The peer reviewers thought, yes, this is it meets everything we want, except we require you to do some revisions. We don't think they're too much that you would have to potentially be outright rejected, but if you do these sets of experiments, or which are limited, and if you address some of our questions, maybe the writing wasn't unclear, or you didn't analyze this properly, or we think you could do that in a reasonable time frame, and so we want you to complete these reviews, respond to our comments, and then return to step 2. In other words, perform those works, do those revisions, send it back to the editor so they can send it back for another round of peer review before final acceptance. And so that takes another significant round of time, depending on how long you have to perform experiments to meet those criterion and then have it go through the review process all over again. The third and the most happy outcome, but a rare one, is outright acceptance. And they said, your work needs just maybe a minor tweak in wording, but otherwise everything is done appropriately, and so we're going to go ahead and publish it as is. That's obviously the most happiest of the trio and cause for celebration, and it's time to party like it's 1999. I know you don't know my references, but if you do know this one, well, keep calm and party like it's 1999, Prince style. Let's move on to our second objective of this particular video, which is understanding sort of the purpose of both what goes on during the peer review process and then as a reader of the published work, how you would actually analyze this. And so overall, it's to improve the science. I'll say that again. We want to make sure we are better scientists, that the science is correct, that the findings are conclusive for us to accept the understanding that is trying to be conveyed before we all move forward and try to build on that work. Everything that's published is a puzzle piece towards understanding a big mechanism, whether that's how cancer works, or how infection works, or anything else that science is achieving. Every author's contribution is one piece of a big puzzle, and we all have to work together as a scientific community and as a readership to vet and do this together. So I always think the purpose is to improve the science, and we want to do that by identifying any potential inconsistencies with the method that you've been taught. So let's break this down into sort of the ways you can do an analysis based on the anatomy of a paper. So the first one we're going to tackle here is related to the materials and methods section. So really kind of vetting the methodology that the researchers used and the design of the study at large and each individual experiment as well. So right away, we're going to start with the materials. This particular section should give you a thorough description of all the solutions and reagents and chemicals that were used to perform the study. And you need to think about them very closely to see if they're aligned with what consistently is being used in your field. One of my favorite examples was an animal study that looked at the role of sugar in the diet in terms of how it modulated bacteria in the intestine and overall animal behavior and physiology. And I looked into the materials and I learned that to get the sugar into the animals, they actually used grape Kool-Aid. And I thought that was a particularly odd choice, but it was the choice they used, and it was not inconsistent with what other groups were doing. So you want to make sure that the materials are consistent, that you understand what they are, and that in some cases journals have provided the manufacturer that they purchase those materials for. And that becomes very, very important for you someday as a user when you might want to repeat a study or build off of a study and know, oh, where did they actually buy those materials? I know that as a reader, we're teaching you to read top to bottom this paper, and so that you understand how to do it. But in practice in your career, you might not actually be doing that in full. You might just be looking for a particular method or a particular material so that you can do your experiment, or just looking at a particular set of results so that you understand one of them. Sometimes you don't have to vet the whole paper. Once you get deep into a particular field, you really don't even need to read the introductions anymore because you're so familiar with them, given that they're very consistent for a particular field. So first, we want to look at the materials and vet them thoroughly. Next, and very importantly, the choice of study model. There could be limitations and or inconsistencies with how the researchers set this up, and are making conclusions they're based on those particular study models. So for example, if a researcher is doing cell culture and using human cells that are in isolation of everything else and put into a dish and grown in media in a lab, and then making a big sweeping conclusion about human behavior, you should slow down and say, well, you are getting there. The human cells are one step of the puzzle, but you grew them in a dish. And so there's limitations between making that sort of statement and a much broader one. And I'm going to return to this in a little bit. Study models might be animal, and maybe your field of study uses mice. And you want to make sure it's consistent with other publications. If they start to use a different model, like a chinchilla, that might not make sense to you, unless, of course, you're studying middle ear infections, in which case chinchilla, believe it or not, is the study model of choice. And if you don't know what a chinchilla model looks like or what a chinchilla is at all, I encourage you to do a little Google image. They're really cute, little furry creatures. So you want to vet the study model, whether that's a benchtop in vitro experiment for community behavior of bacteria cells or anything else you're doing. You then want to look and make sure the sample size is not only consistent with your field, but also consistent within a paper, and especially consistent within a particular figure. There are obvious reasons sometimes that a sample size might vary. So for example, you started off with 6 mice and in group 1, and then you had 6 mice in group 2. Let's return to the week 3 biomath example where you had treated and non-treated animals. And maybe the treated group, 1 died off, and so you went from 6 to 5, but it's still a significant amount of animals to run with the final result. You might see it vary then from comparing 6 to 5. Not apples to apples, but we can roll with it. Sometimes it's very inconsistent and you can't. So you want to make sure that there are a correct number of biological and technical replicates, and we're going to teach you about what those mean both this week and in subsequent weeks, and that it's consistent with your field. So you have to evaluate the sample size. You also want to make sure that the study was designed with all the appropriate controls, and that they are represented in the data set at a satisfactory manner so that you can actually believe in the results. Controls, both positive and negative controls, are extremely very important. A well-controlled experiment gives you the trust that you need to think that the result is one that you can build off of or not. For example, let's go back to our treatment versus non-treatment of the mice. Let's say we were treating with compound A, and that's our treatment group. Well, compound A may have been dissolved in a buffer, let's say ethanol. So compound A was dissolved in ethanol and given to the treatment mice. The vehicle control for this experiment, the negative control, would be ethanol by itself. We want to make sure that the compound that we're testing, compound A, is giving the effect that we're looking for on the animal one way or the other, and not the thing we diluted it in. So ethanol in that case would be a control. The second component of the paper, the anatomy you've learned that we really need to vet thoroughly, is the results. And there's a lot of ways to do this, just like there are the methods, and we're only giving you a couple. And one of the key ones that I like to start with, especially at this stage, when you're learning how to do this, is looking for agreement or lack of agreement between what the sentence says in the results section and then what is actually represented and shown in a particular data set, whether that be a figure or a table. Believe it or not, there are inconsistencies, and we're going to task you with finding some on your own this week as you're analyzing the paper of choice. So in other words, let's say a sentence says that a result is significantly different between two groups. And that's what the result says in the sentence, okay? And then it references, let's say, figure 1. So you turn your attention to the data, and you want to look for an asterisk with a p-value indicating statistical significance. If it says that the two groups are not different or they're the same, then you should see a lack of statistical significance. Sometimes the sentences don't match, or sometimes, believe it or not, the sentence will say the result shows an increase, and then you look at the data and it was actually a decrease. This happens too. So you want to look for agreement. And then, as a bonus, you kind of want to look to see if there's anything else in the data that might not look consistent. For example, they might want you to look at the vehicle control versus the treatment, and let's say it goes up significantly, and they say, oh, great, it's significantly increased. And then they do a knockdown treatment, and it goes down significantly from the increase. But it's still significantly different than the vehicle control. Those differences between the knockdown and the vehicle control could also be important. And that's not necessarily a deal breaker for you to believe in the work, but that could also lead you to new questions that generate new experiments for future directions. We want to know now, why is the knockdown not quite the same as the vehicle control? Maybe there's an important difference there too to understand. And I think that evaluating the results to do this in part, which is to identify future areas of study, is a really important tool for you to learn. And I think it actually makes science more accessible. I always thought that to be a successful researcher and scientist, I would have to develop Nobel Prize-worthy ideas and questions and hypotheses. And while there's absolutely a certain element of designing your own, I then learned and took a lot of comfort in the fact that the literature, the papers, the results provide a needed framework from which to springboard from. And so I wasn't locked in some room all by myself trying to think of all this by myself. I was actually using the papers to generate a springboard, a platform from which to draw new experiments, new aims, and new directions, and build off of things that I thought, wow, that might not be a critique necessarily or a fault with the study that I'm analyzing this paper, but this could be a direction that they might not be thinking about going in that we could take our work. And so it actually made it feel a lot more accessible to me. You always want to look at the axes / the scale on a graph, or any other way that the particular results are presented to you, any other parameter in a data table or otherwise. And so the first thing I always do is ask myself, what's on the y-axis and what's on the x-axis? What are those scales showing me? Is it percent increase? Is it value A versus value B? Is the x-axis at a time scale? Am I looking at, let's say, growth of bacteria treated and untreated by a compound? in a test tube over a course of 12 hours. What am I actually looking at? Let me understand the results, and let me make sure that these axes, scales, or other parameters are consistent with how other biological studies in my particular field are done. So for example, if you do a growth curve of bacteria, and let's say the majority of the field does this over a course of 24 hours, but this group does it over a course of only 10, you might be left to conclude that the authors should continue the study, redo the experiment, and complete the full 24 hours to be in alignment with everybody else. This takes a lot of time to realize the other works in the field, so don't be afraid that you didn't know that or anything else related to any particular field. Once you're in there, you'll start to feel that comfort level increase. You want to make sure that the data are visualized appropriately, and we're going to have a whole week on this to come, and you're going to learn how to do this, and you've kind of thought about it a little bit already, but you want to make sure that, let's say, bars or individual data sets, numbers of things are consistent and presented in an appropriate manner that aligns with everything else that you know to be true within a field. And of course, that the choice of statistics is appropriate, and the way that the data is analyzed to get those statistics was correct, whether that was an ANOVA or a t-test or any of the other ones you've learned. You could fold this into the methods as well. We could have vetted this there, and that would have been just fine too. Or you could vet it within each individual result. A good result will potentially tell you the way they did the stats in the figure legend, as well as the method section. So there should be some helpful redundancy, I like to say, in the choice of the stats and the way the data was visualized, both within the methods and within the figure legend of the results themselves. So you can always make sure that those are done correctly. In other words, did they correctly choose a one-tailed or a two-tailed t-test, or should this have been a completely different method of analysis altogether? While the majority of peer review for publication and for you as a reader to vet scientifically in your analysis of an article, while the majority is done in that first two components, the materials and methods sections and the results, we also want to consider whether or not the introduction and discussion sections also comply. And so this becomes a little bit more broad than the nuts and bolts of sort of experimental design and what a graph is showing you, and instead becomes an exercise in placement within the literature that's been established for a particular field. And so a good introduction and a good discussion have enough references to other works to do that contextualization. And so you want to make sure you're citing supporting evidence and sometimes conflicting evidences that place your work within the correct context. It's important for a lot of reasons. We want to make sure that we understand the novelty or lack thereof necessarily of a findings. We want to acknowledge the previous works that are done by other groups that have led to yours. As you learned when reading the paper, a very good introduction section sort of establishes the significance of the work and then references the previously done work that led to your question. And so you get a feel for all those things. And a very good discussion will then contextualize their results with a nod to the field in terms of where the work could go into the future. So you want to make sure you've done all those things. And I always give a little plug here for referencing methods in particular. I think we always think to reference sort of the idea or the result, but sometimes not as much the method. And sometimes people will just reference a paper in which the method was referenced from a different paper, and then you go down this reference rabbit hole trying to find the original place the method was referenced, or I should say actually established. And so try to go all the way back and find that first establishment of a method and reference that one, instead of a paper that came afterwards referencing that. That will be very, very helpful to any reader. We also want to make sure that the wording is consistent. You oftentimes in science find things that are overstated. Remember that a study done in mice is indicating results for mice, not humans. So make sure your understanding and appreciation for where it is appropriate. This was a hard lesson for me in graduate school when I did this experiment looking at gallstones and salmonella in a test tube, and I thought I had cured all humanity of a particular disease, and my advisor said, "Yo, you just did something in a test tube." There's a lot of steps between this and human beings and clinical trials and everything else that would go with that. So just know where the work was done and whether a result is suggestive of something, and we're going to go into specific wording in the next video, but or whether it's actually conclusively indicating something. And importantly, focusing on correlation versus causation, making sure authors are using the correct wording to describe their results. We're going to go into this further in the next video as well. And remember, if all this is done, this should be happy, this should be joyous, and this is for the whole global community of scientists and non-scientists alike. I always believe firmly that the job of a biologist is to communicate the results with the scientific community, but also with the non-scientific community. An appreciation of your work at both of those levels is not easy to do, but is necessary to make the scientific enterprise continue, so that grants are funded and papers are published. And in your discussions in class, your instructors might potentially start to tell you a little bit about sort of the different payment models for these papers becoming published, what a journal subscription fee looks like versus open access or not. And those things become very, very important when you think about getting this out there to the planet. So this concludes the first of the videos this week, and please feel free to move on to the assignment that's associated with it. Thank you.